Bismillahirrahmanirrahim. We are here to start chapter number 1 of pathology, that is cell injury, cell death and adaptation. We know in a normal cell there is a hemostasis. Adopted cell is that type of cell that is in between normal cell and an injured cell. For example, if a cell is under stress, we say that it is an adopted cell. If it is not able to adopt the cellular adaptation, it will result in cell injury. Cell injury occurs in a normal cell in response to the injurious stimuli, or it can occur when a stress cell, that is adopted cell, gets inability to adopt. Cell injury can be either reversible injury or irreversible injury. When it is severe or it is progressive, it is irreversible injury and it results in cell death, that is either necrosis, or apoptosis, that is programmed cell death. When cell injury is mild or transient, it can be reversed. Okay, let's talk about this adopted cell. Adopted cell is stress cell, and this is in between the normal cell and injured cell. Stress cell is always reversible, that is stress cell is reversible cell. Yes. Irreversible injured cell always goes to necrotic cell. Let's talk about this injured cell, that is cell injury. We say that if we talk about the injured cell, we say that this injured cell can be either an inflamed cell. When you talk about inflammation, what is inflammation? Inflammation is body vascular, either you talk about the vascular, either it is neurological and cellular response to injurious stimuli. So inflammation is defined as the body vascular, neurological and cellular response to injurious stimuli. This cell, injured cell, can be either inflamed cell or it can be the necrotic cell, what we are going to discuss in the subsequent topics, necrosis. And then comes the dead cell. What do you mean by the term dead cell? Dead cell is the fixed cell. When voluntarily you dead the cell by using the 10% formalin, and step 1 is the denaturation. So this was about the introduction of the cell injury, cell death and adaptation. Let's talk about the adopted cell. Adopted cell is having the 4 types. It is always reversible with the exception of one condition that I will explain, that is called dysplastic cell, that is malignant cell. Now what happened? That it is having 4 types, that is atrophic cell, hypertrophic cell, hyperplastic cell and metaplastic cell. Metaplastic cell is having further 2 types. One is called as epithelial metaplastic cell and the second one is mesenchymal metaplastic cell or stromal metaplastic cell, and we are going to discuss it in the next videos. Okay. First, let's talk about the first cellular adaptation, that is cellular atrophy. Cellular atrophy is the shrinkage in size of cell by loss of cell substance. How it is defined? It is defined as a shrinkage in size of cell by loss of cell substance. We know cells are having a trilamina structure, that is bilayered. In atrophy, you will see that over there histone condensation is seen. Mechanism is that either protein synthesis is decreased or there is increased protein degradation. If you talk about the decreased protein synthesis, it is due to reduced metabolic activity, and increased protein degradation is due to the ubiquitin proteasome pathway. What do you mean by the ubiquitin? Ubiquitin is a stress protein. It is having 76 amino acid, and while talking about its molecular weight, we see it is having 8 Dalton molecular weight. Now, how increased protein degradation occur? Nutrient deficiencies will activate the ubiquitin ligase. It is an enzyme. What it will do? It will attach to the small peptide ubiquitin, and as a result of this, it will attach this ubiquitin to cellular proteins. Then it will target them, and then as a result of this, it will lead to the degradation in the proteasome. So that's why it is called as ubiquitin proteasome pathway, because ubiquitin is attached to the cellular proteins, and after targeted them, they are degraded in the proteasome. This proteolysis is also seen in cachexia, which we have discussed in neoplasia chapter, that it is the loss of the body fats. So as a result of this, excessive weight loss occurs. Third point is autophagy, formation of autophagic vacuoles. Then comes during cell cycle. What happened during cell cycle? Progression through cell cycle, that is UNS transition, is regulated by the cyclin, and along with that we are having the enzyme that is called as cyclin dependent kinase. Now cyclin dependent kinases are activated by binding to the cyclin. They phosphorylate different proteins, and cyclins are of 15 types like D, E, A and B. Now what happened when the cyclin and cyclin dependent kinase complex combine together, they phosphorylate the target proteins that drive the cell through cell cycle. In order to check the cyclin and cyclin dependent kinase complex, we are having cyclin dependent kinase inhibitors. They act as the checkpoint. One is G1S and another is G2M. Means during DNA replication and after the mitosis, there are the checkpoints. If DNA damage is there, then cell will either show the apoptosis, or according to the P53, that is the genome of the cell, guardian of the cell, it will P53 will try to show senescence. So this is how the cells are removed, either by apoptosis or by senescence. Okay, now let's talk about atrophy. Atrophy can be physiological, it can be pathological. Physiological, for example, hormone depletion in menopause, or pathological, for example, denervation. Examples include breast atrophy, uterus atrophy, myometrial atrophy, and endometrial atrophy. Both are included in uterus atrophy. Testicular atrophy, brain atrophy, for example, in Alzheimer's disease, muscle atrophy and glandular atrophy are the examples. Causes can be decreased workload, denervation, immobilization, loss of hormone, inadequate blood supply, inadequate nutrition, and aging, that is called as senile atrophy. As we have discussed, atrophy, that was the first cellular adaptation. Let's move toward hypertrophy. Hypertrophy occurs when cell has limited capacity to divide. Definition is, it is increase in size of cell resulting in increased size of organ. Either substance of cell structure is increased, or either there is polyploidy, diploidy and monoploidy. Mechanism is, it is having two types of mechanism. One is called as the mechanical trigger and other one is called as the trophic trigger. Mechanical trigger occurs either due to stretch, and trophic trigger is due to the soluble mediators that stimulate the cell growth, such as growth factors and adrenergic hormones. So these stimulation in turn, what will happen as a result of this trophic trigger, they will lead to the signal transduction pathway. They will turn on the signal transduction pathway, and that will lead to the induction of number of genes. Once the genes are activated, they will stimulate the synthesis of cellular proteins, including growth factors and structure proteins. More proteins and myofilaments per cells, increased force generated with each contraction, and as a result of this, over here you will see the increased work demand. As I told you, that is due to increased adrenaline and noradrenaline, increased stress. So there would be increase in size of chamber of heart if you're discussing it in heart. There is also switch of contractile protein. What do you mean by this term, switch of contractile protein? There is a switch of contractile protein, for example, from adult to neonatal form. For example, in muscle hypertrophy, alpha myosin heavy chain is replaced by beta myosin heavy chain. In mechanical trigger, example is, for example, increased BP will cause the distension of blood vessels and it will lead to the muscle hypertrophy. Causes can be physiological as well as pathological. Physiological, for example, enlargement of uterus during pregnancy. Also, it is seen in striated muscle cell. Striated muscle cell involves skeletal muscle and heart muscle. Skeletal is having multiple nuclei while heart is having single nucleus. Pathological involves cardiac enlargement due to hypertension or aortic wall disease. Common organs may be heart, skeletal muscle, uterus, but it is also seen in breast and prostate. Okay. Normally, if you see here in gross morphology in heart, if hypertrophy occur in heart, for example, in endocardium, in it, as a result of this, there would be endocardium thickness, our interventricular septum size will increase, and valve will become incontinent, that is loss of voluntary control. It is clear from here that heart is having a single nucleus, around its syncytium is present. This is the normal hypertrophy mechanism. If trophic trigger is there and there is increased stress, that is adaptation to stress occur, and it can progress to functionally significant cell injury if the stress is not reduced. For example, if the stress is not reduced over here, then as a result of this, what would happen? Then there is increased burden on heart, and as a result there would be degenerative changes in heart. The net result would be ventricular dilatation and cardiac failure. So this will definitely clear you that adaptation to stress can progress to functional significant cell injury if stress is not reduced. Like over here in the complications, you will also see that cell will be having fragmentation, loss of myofibrillary contractile element, polyploidy is seen, fibrosis is seen, and fat cell is also seen in the heart. Let's talk about